Thank you for this very kind invitation. So we'll present some tools that we are developing to optimize, to try to optimize blood transfusion, red blood cell transfusion. So as you know, each year about 85 million units of red blood cells are transfused worldwide, and there is two problems. First is that despite extensive serological and molecular typing, and despite the presence of experts like Thierry, at least in France, there is still significant prevalence of alloimmunization. In France, this prevalence is about 3% in the general population, and it can reach 8% in the hospitalized population, and this alloimmunization results in transfusion accident, which can be sometimes fatal. And also, if you consider blood transfusion efficiency, storage lesions, which are membrane alteration like membrane lipid and protein oxidation, shedding of toxic microparticles or Bain-3 modification, for instance, these storage lesions, which occur during blood banking, are thought to be responsible for the removal of up to 25% of the stored red blood cells during the first 24 hours after transfusion. So our objective was to try to improve the safety and the efficiency of blood transfusion. First, by developing new tools for a better characterization of both the blood group antigens and antibodies. One approach is, as presented by Thierry, the serological and molecular characterization of new blood group system, antigens and alleles involved in these blood group incompatibilities, but also to develop new tools for red cells immunophenotyping, and I will present some data on the production of nanobodies or VHH antibodies, and also new tools for allo or auto-antibodies characterization in complex serum by the use of recombinant blood group antigens. And to improve the efficiency of blood transfusion, we present some data which have been initiated in the group of Olivier Hermine, and now in collaboration with our team in ENTS, the development of tools to counter the red blood cell storage lesion during blood banking, and we'll show you some results obtained with the use of serotonin as an antioxidant in mouse and human models. So first, new tools for red cell immunophenotyping. As you know, there is a lack of potent antibodies against some blood group specificity, like the RHC composite antigens and the antigens of the KNOP and COST system. So Olivier Bertrand in our lab decided some years ago to develop EV chain only, or VHH, or nanobodies. These EV chains only nanobodies, or VHH, are very simple antibodies which are produced in camelidae, dromedary or llama, and contain only VHH chains. And as compared to conventional antibodies, these VHH or EV chain only antibodies are like conventional antibodies, they are target specificity, high affinity, but they have the advantage to be highly stable, they are easy to manufacture and engineer. They can access, because they are very small, to enzyme active site or receptor cleft, and they exhibit a low immunogenicity. So it's quite easy to obtain a cDNA library of this VHH. If you have a dromedary or llama, and we found these guys in Canary Island, not in France, in Canary Island, and these camelidae are immunized either by a different type of immunogen, and the cDNA for the VHH are amplified by PCR, inserted in phage-mid, and these phage-mid are screened by the antigens of interest. So now in the lab, Olivier and colleagues have immunized 4 dromedaries with different type of antigens, either DNA fragment expressed in E. coli, like DARK for Duffy antigens, RAG for Rh antigen associated glycoprotein, but also other protein like a protein of Plasmodium vivax or HIV. The antigen can be also expressed in eukaryotic cells before immunization, in insect cells, but more interestingly, also we can immunize this dromedary with human-packed red cells. And these two dromedary have been transfused by the whole human red cell unit. They felt rather bad for 10 to 15 minutes, but after everything was okay, and they produce very nice antibodies. So today we have in the lab about 100 nanobodies which have been isolated. Some recognize the DARK antigen of the Duffy blood group system. These antibodies have been used to inhibit the function of the Duffy antigen, the chemokine binding function, but also the receptor for Plasmodium vivax properties. Several recognize glycoprotein A, which is very abundant in human red cells, so it's not surprising to have a lot of MN anti-blood group VHH antibodies. And that can be used for diagnostic application, like blood typing. Several recognize BCAM or ICAM4 molecule, which carries the lutein and LW antigens. And these molecules are very important involved in sickle cell disease. And at least the antibody against BCAM has been found to be able to interfere with red cell adhesion to endothelium in red blood cell from sickle cell disease patient. And we have now two libraries of 9, 10 to 8 phage-mid, which are ready to screen for blood group antigen and other red cell membrane protein. And of course, these two libraries are available for collaborative projects if you are interested. The second way to increase the transfusion security is to produce recombinant blood group antigens for the detection of antibodies against low-frequency blood group antigens in complex serum. So as compared to the use of reagent red cells, and I remember that some phenotypes are very rare, and to type some antigen antibodies, the use of recombinant antigens will avoid limitation due to low frequency or low expression levels of the blood group antigens recognized by alloantibodies present in complex patient serum. They will be recognized by a single antibody in this complex serum, and they should be easy to use because they don't need enzyme treatment, no immunoabsorption needed for antibody characterization. The major problem is that it's very difficult to obtain high-level expression and to purify recombinant multitopic membrane protein, like the Rh protein or the KID protein, for instance. Rh pass 10 times through the red cell membrane, KID 7 times. I will give you the example of the Rh and RAG protein. So they both belong to the Rh system. The Rh antigens by themselves, the D, C, and E and CE antigens. There is now 54 Rh antigens. The molecular bases have been elucidated in our lab, but also by others. Most of them have been elucidated. So it's not possible to express many alleles of the Rh antigen in heterologous system. RAG, the Rh-associated glycoprotein, has been shown more recently to carry at least 4 blood group antigens, all are RAG4, Duclos, and DSLK. And the molecular basis is also elucidated, and so it's also possible to express all these alleles in heterologous system. The problem is that we and others try to express the Rh, the erythroid Rh protein, Rh or RAG, in eukaryotic cells. We only obtain very low membrane expression, as shown here. And as here in contrast, when we try to express the non-erythroid Rh homologue, like RHCG, we obtain a very high expression level of expression. So why it's difficult to obtain a high-level expression of RH and RAG? Certainly because, as we have shown quite a long time ago, RH and RAG belong to a very large macrocomplex in the red cell membrane. And to obtain high expression of RH and RAG, we need to reconstitute this complex in recombinant cells, and this is very difficult. So, sorry, it's in French. Okay, so we have decided to use acellular or cell-free system to produce these RH antigens. So here are very preliminary results which have been obtained in the lab with a cell-free system. I'm sorry I cannot go too far in detail because we have a patent under submission and I'm not allowed to speak too much, but just to show you the first result to obtain. And for the first time in this cellular system, we obtain a very high yield of purified RAG protein. You can see here for the first time in glucomacid gels the RAG protein, which high yield with a high purity. So ongoing experiment, we are now producing this acellular system also the RHD and RHC antigens and the KID, which is carried by the UTB protein. And also we try to refold this protein in native structure because most of the antibodies recognize nonlinear epitopes, so we have to reconstitute this protein in their native three-dimensional structure. Again, sorry, we have a way to do that, but I cannot tell you which one, maybe next year if you invite me. So the third part of my talk, concerns the improvement of the efficiency of blood transfusion. And I will show you some results on the use of serotonin, which is a well-known neurotransmitter, to counter the storage lesion. So this story began in the lab of Olivier Hermine. So what was the rationale to study the role of serotonin in red blood cell survival? In fact, this began with a mouse story, and people in the lab of Olivier wanted to make knockout mice for serotonin to obtain a nervous breakdown model. And in fact, these mice were not depressed, but were anemic. And the reason why is that they didn't know that there is at that time that there are two genes for the expression of serotonin. One, THP2, which is responsible for the expression of serotonin in the central nervous system, and one for expression in periphery. And when they perform the knockout, and in fact they perform knockout of the gene responsible for expression of serotonin in periphery, but not in the central nervous system. So it's why the mouse are not depressed, no burnout. But they have no serotonin in the periphery, it's why they are anemic. So the author shows that these mice display a phenotype of a typical phenotype of regenerative macrocytic anemia, which indicate that serotonin is necessary for normal erythropoiesis. Here they show that in vitro, with mouse red blood cells, incubation of mouse red blood cells with serotonin protect from hemolysis in a dose-dependent fashion. And in a mouse model of blood transfusion, Pascal Amiro, I've shown here in this experiment, he took a wild-type donor, he took blood, and after deleukocytation, this blood was supplemented or not with serotonin. This blood was banked at 4 degrees up to 14 days, which is equivalent of 14 days of 80 days in human. And after this red blood cells incubated with serotonin and not incubated were transfused to the receiver. And you can see here that incubation of these red blood cells with serotonin prior transfusion result in a higher, in less post-transfusional hemolysis. So this shows that serotonin supplementation can improve the 24 hours post-transfusion red blood cell survival. In human, in fact, only experiment in vitro has been performed now, and in tube, with a low hematocrit, they also observe that serotonin they observe a serotonin protective effect on the viability of human red blood cells, at least at low hematocrit. So what are the mechanisms of this protection? I don't know if you see something.
No.
Okay.
I'm seeing nothing.
Okay.
Yeah, but something's with the light.
No, okay.
Just tell us what you could see.
Okay, first, Slim Azouzi in the lab have shown that serotonin has an antioxidant, maybe antioxidant effect by incubating red blood cells with hydroxyperoximane, which is an oxidant. Here you can see that the control red cells in the green channel, they are not oxidized. When these red cells are incubated with this oxidant hydroperoximane, you can see here normally high yield of membrane oxidation. And when these red cells are incubated with the oxidant plus serotonin, there is no more oxidation. So this means that serotonin is able to protect red blood cells from oxidation. But the mechanism is not very clear because there is no serotonin receptor in mouse and in human red blood cells. So Slim makes the hypothesis that maybe serotonin is only partitioned into lipid bilayer without the need of receptors. So here I have a movie, but I think. Okay, so maybe. No, it doesn't work. Okay. In this movie, you can. Okay, yes. This is molecular dynamic in silico experiment, and you can see that serotonin here stay at the surface of the lipid bilayer and is blocked at the polar end of the lipid. And by wet lab experiment, Slim also shown that serotonin is able to capture growth at the membrane surface. So the antioxidant activity of serotonin takes place exclusively at the membrane interface, either through the interception of diffusing ROS species or the termination of lipid peroxide radical by interacting with oxidized lipid. So in conclusion, serotonin reduced half-life in knockout mice. There is a reduced half-life in knockout mice, a reduced half-life of red blood cell when transfused to knockout deficient recipient. Supplementation of serotonin is able to retard RBC hemolysis in vitro, and serotonin supplementation increase red blood cell post-transfusion survival in a mouse model. And in human, serotonin is able to increase red blood cell viability in vitro and to prevent membrane oxidation. So the challenge now is the real life and to determine if serotonin is able to protect red blood cell in bag. And to be honest, we have observed that serotonin is able to protect red blood cell in vitro only at low hematocrit, but not in blood bags. So we think that maybe we are now screen for other antioxidant molecule, which we hopefully will be able to protect oxidation in blood bag and also at a very high hematocrit. Thank you.